Hello everyone, my name is Yatharth Kakkad and I'm from Sarasota, Florida in the United States. I researched intracranial aneurysms, which are a life-threatening vascular abnormality that affect 168 million people worldwide. Of these affected individuals, there's a disproportionately high 45% mortality rate after the aneurysm ruptures in their brains. The problem, however, is that the mechanisms by which these intracranial aneurysms develop remain largely unknown. Thus, my project investigates a novel mutation, the E736D substitution in the protein sequence of COL22A1, a type of collagen. The E736D mutation compromises the ability of COL22A1 to maintain the stability of blood vessels. This increases the risk of developing intracranial aneurysms. My project generated a novel line of transgenic zebrafish embryos capable of expressing the mutated COL22A1 protein via the binary Gal4 UAS system. Compared to the primary and secondary controls, this novel mutant line demonstrated the strongest hemorrhagic phenotype ever recorded and also allowed for greater experimental modularity, thus significantly cutting costs for fellow researchers. I also quantified the presence of a cell called pericytes in the brains of my novel mutants. Previous research has indicated that decreased vascular stability correlates with decreased pericyte coverage, though this trend wasn't observable among the E736D embryos. Finally, I used live confocal imaging and specialized software to create 3D reconstructions of the cranial vasculature of my novel mutants. Data analysis revealed that E736D embryos developed significantly compromised vascular networks, characterized by reduced branch points, vessel length, surface area, and volume. This means that tissue scale impacts are now viable as a benchmark for other researchers to evaluate results.